Well, thank you all for coming. It's my pleasure to introduce Jenny Fisher as today's seminar speaker. Jenny is currently a senior lecturer at the Schools of Earth and Environmental Sciences and Chemistry at the School of Earth and Environmental Sciences and the School of Chemistry at the University of Wollongong in Wollongong, Australia, which is just south of Sydney, for those of you who don't know. But Jenny is an American. She went to undergrad at Caltech, and then did her PhD at Harvard with Danielle Jacobs' group, during which time I met her in Alaska during the Arctas campaign. And then she, after graduation, moved to Wollongong to do a postdoc in the School of Chemistry in 2012. And so she's been there since then and seems well on her way to staying there for at least a while. Last year, she received the L'Oreal UNESCO Fellowship for Women in Science in Australia, and she has a wonderful video on her homepage that you should go look at, where she talks about her research and the fellowship. She's also a member of the GEOS-Chem steering committee, and is the co-chair for Mercury and POP's working group. And she's been here for the past week and will be here for a few more days. And we're working on designing an experiment in Australia called Koala that she'll tell us about at the end.
All right, thank you for the introduction and the invitation, Louisa, and thanks everyone for coming. I am a modeler, but I have benefited from a very large number of observations made by a lot of really collegial colleagues. So there are a lot of names listed here, and this isn't everyone, but I just want to make sure I acknowledge that I can't do the work I do without those collaborations. I also want to thank a number of different groups and agencies for computational and funding resources. So I was warned that there may be some non-chemists in the room, so before I delve into the chemistry, I just wanted to take one step back and explain a little bit about why I'm doing the work that I'm doing. And while I'm looking at organic nitrates, I'm really interested in how they impact nitrogen oxides. And nitrogen oxides are the sum of NO and NO2, and these are central to all sorts of things that we care about in atmospheric chemistry and composition. I really love this figure from this review paper about the nitrogen cycle, just because it shows how complicated all of this chemistry is and how many different things are involved. And these nitrogen oxides impact OH, which is the cleansing agent for the atmosphere that impacts our oxidizing capacity and our methane lifetime. Ozone, which can be a pollutant at the surface or a radiative forcer in the free troposphere. Nitric acid, which affects nitrogen deposition and acid rain. And secondary aerosols of various forms, which also impact pollution, radiative forcing, nitrogen deposition. So it's kind of central to all of these different processes. And it's complicated. And what I've been particularly interested in over the last couple of years is these species called alkyl nitrates, which are of the form RONO2 here. And these are species that are formed when we have a reaction between a volatile organic compound that can be from a biogenic source, like isoprene or monoterpenes from trees and terrestrial vegetation, or an anthropogenic VOC that can be from something like methane or ethane from oil and gas extraction. And when that is emitted to the atmosphere in the presence of NOx, there are two different options for the chemistry. So the main pathway is this one, which isn't particularly interesting from a NOx budget perspective because it just returns that NOx back to the atmosphere. But this is a minor channel, something like 1 to 5%, depending on which species we're looking at. And that forms this alkyl nitrate. And that's interesting to the NOx budget because that can then go on to do a variety of different things. So one option is that if it's sticky enough, it can be deposited right away, and that can be a source of nitrogen to deposition to ecosystems. It can undergo standard atmospheric chemistry, photolysis, oxidation, and return that NOx back to the atmosphere. It can be taken up to the aerosol phase to form a particulate, which itself can be deposited or can form nitric acid to be deposited. Or either this gas phase species or this particulate species can be exported so that these loss processes are happening somewhere different from where the formation is happening. And the reason that this matters for the NOx budget is that if we go through this channel, we'll recycle the NOx back to the atmosphere. But if we go through any of these other channels, then this becomes a terminal sink for NOx, and so can influence how much NOx we have, either in a source region or somewhere else where it's been exported. So the goal of what I've been doing recently is to try to quantify the impact of all of this on understanding the NOx budget. And I'm going to talk today about two different bits of work we've been doing in this space. The first one is looking at these biogenic species, so isoprene and monoterpenes, and what happens when you form alkyl nitrates from these, using some data from the Southeast US. And the second one is looking at more of these anthropogenic species, so ethane, methane, and propane, and looking at global scale implications. Okay, so part one. So one of the things that I was able to be involved in was the 2013 NASA Seekers aircraft campaign. And this took place in August and September of 2013, flying out of Houston, Texas, but over all the regions that are covered with these black flight tracks. The base map here is a plot of isoprene concentration in surface air. So the idea was to try to understand the unique chemistry that happens, or one of the ideas of the campaign was to understand the unique chemistry that happens in these very high biogenic source regions. And we covered a lot of regions with high isoprene and some regions with lower isoprene, and were able to get a pretty good look at what was going on in that region. In addition to the flight campaign, we also had a ground site based at Alabama called SOAS. And the reason that this campaign was really a unique opportunity to try to answer some of these questions is that there were a lot of really exciting instruments on the plane. I don't need you to pay too much attention to all the details of this chemical flow here, but this is basically the chemistry that happens when isoprene is oxidized in daytime. And the important thing is that everything that you see in green was something that was measured on the plane. And this gives us a really nice set of constraints to look at what do we have in our current representations of this chemistry in the model, and how well does it actually track all these different pathways and all these different possible products. So to do that, I've been using the GEOS-Chem chemical transport model, and I was using it in our newest 25-kilometer resolution over North America. This detail is just for any modeling nerds who happen to be in the room. Don't worry about it, except to say that our base BVOC emissions are from Megan, except that we have reduced isoprene a little bit and increased monoterpenes to match what we saw on the plane with the PTRMS. We've also reduced the NOx emissions from NEI 2011 based on work that Katie Travis from Harvard has published. And we put in a lot of different updates to the isoprene chemistry and isoprene reactions and monoterpene chemistry as well. This plot here is just showing isoprene emissions over this region in the model, and that orange box is the southeast, as I'll show averages over for the rest of these slides. Okay, so the first thing I wanted to do was just see how well is this chemistry represented in the model, and in particular for my interest in the pathway that goes through alkyl nitrate formation, so reaction with NOx. So I'm going to show a bunch of these profiles, and these are all vertical profiles from the aircraft. The black lines are from the observations, the red lines are from the model, the horizontal bars are the variability across that southeast region, and the gray shading is the experimental uncertainty on the observations. So this species here is isoprene nitrate, which is the very first one that you form after oxidation by OH and then reaction with NO. And you see we have quite good agreement over the course of the campaign. If we step through to some of the second generation products, so these are things that form from this isoprene nitrate under further chemistry, we have a number of different species that were also measured on the plane. They also give quite good agreement within the experimental uncertainties of the model. So we're reasonably happy with this simulation of these alkyl nitrate species. The one problem that remains is that there was another measurement on the plane of organic nitrates or alkyl nitrates, and that's this black line here, which represents their total as measured by the TDLIF. So that measurement can't distinguish these individual species. And when you sum up all the individual species, which is sort of what we've got in all of these lines here, don't worry too much about the detail, you don't get the same thing as when you measure the total. And that was a little bit of a surprise because this hasn't been true in previous campaigns where both things have been measured. It's unclear whether there are experimental issues to work out here or whether there are a bunch of alkyl nitrate species that we aren't yet able to measure individually that are affecting the chemistry in the Southeast US. So this remains a bit of an open question at the moment. But for now, we're just worrying about focusing on the things that we know we can measure and a couple of other species. So one thing we can look at is taking all the species that we know from the observations and all the things that we model explicitly, what's actually contributing to the overall burden budget that we have in the Southeast US. And that's shown here in these colored wedges for the observations and for the model. I'll just highlight a few things. So the first thing is that these orange contributions on both sides represent the total amount of nitrates coming from isoprene, either first or second generation. And that explains about 30 to 50% of what we see in both the observations and the model. This blue wedge over here is monoterpene nitrates, which are less important than the isoprene in this part of the world. They weren't measured on the plane, so we don't have an observational constraint on that. But we can anticipate that that would be a more important source in some regions where monoterpenes are higher, so places like Boreal Canada or possibly Australia. And that's something we haven't previously included in our model at all. So this, I think, is something that we need to think carefully about. The other interesting bit here is the gray wedge, which is the organic nitrate in the particle form. And this, again, is something that we hadn't considered previously, but is anywhere between 10 and 20% of the total. So this is sort of a picture of what we think is going on in the Southeast US. But if you'll remember, my interest isn't particularly in narrowing these things down, which is all right, but not that useful on its own, but really in seeing what this means for our understanding of the NOx budget in this part of the world. So to look at that, what we've done is look at what the ultimate fate is for these different nitrate species, and that will depend on which species we're looking at, what their chemistry is, how sticky they are. But basically taking all of these things together, using the model to portion out their sinks, we find that about 60% of the gas phase species are being lost to this uptake by the particles, and another 15% by dry deposition. And if we look at that in terms of sort of those processes that recycle NOx back to the atmosphere versus those processes that are terminal sinks for NOx, we're finding about 75% of the RONO2 that we form is serving as a sink for NOx. And if I frame that slightly differently and say how much of the NOx that's emitted goes through this process and is lost via one of these processes, then we find that about 20% of the NOx overall in the Southeast US is being lost by forming RONO2 that is then terminally removed from the atmosphere. So that's a reasonable number, but what that number doesn't really tell us is how much variability is there in these regions where we know we have some areas with very high isoprene, some areas with very low isoprene. So what I've done and what I'll show on the next slide is basically calculating this same percent, this fraction of the emitted NOx that's lost through this processing, and mapping that out on a grid box by grid box scale. So that's shown here, and you can see that the importance of this process as a NOx sink is really, really variable, going from 0% in some of these sort of anthropogenic regions up to 100% in places like the Ozarks, which we know to be a very large source of isoprene. This plot on the right is just the ratio of the isoprene and monoterpene emissions to the NOx emissions. So you can see that they're very closely correlated. In other words, where we have very high isoprene or monoterpene emissions, this sink is very important for the NOx, but where these emissions are low and the NOx emissions are high, this sink is negligible. And we can look at that in a slightly different form, and this is a slightly complicated figure, so I'll walk through it a little bit. So the x-axis here in the gray bars are showing cumulative NOx emissions in the Southeast US region. So basically adding up finding all the grid boxes with the lowest 5% of NOx, and then the grid boxes with the next 5% of NOx, and stepping up along that way. And then in the blue, averaging for those particular regions, how important that NOx sink is. So if we look, this is consistent with the previous plot. So if we look at the places where we know we have very low NOx, where that ratio of BVOC to NOx emissions is high, then this sink is really important. It's something like 60%. But what's interesting here is how quickly that importance drops off. And once we get down to even sort of average NOx emissions for this region, we're in the sort of 10 to 15% of the NOx sink. And the reason that that's relevant is that there's been some suggestion that as we move into regimes where we decrease our NOx emissions more and more to comply with air quality regulations, we're going to greatly increase the importance of these alkyl nitrates and these biogenically derived alkyl nitrates to the NOx budget. But actually, this is showing that that might not be the case for quite some time. So if we think about where we're likely to reduce NOx emissions, it's going to be in the places where we already have a lot of NOx, right? So it's going to be improving the cars or decreasing, cleaning up our fossil fuel power plants, whatever. And moving along this axis in this direction, it takes a very long time before that these alkyl nitrates become an important part of the sink. So that's more or less where things stand for the biogenic alkyl nitrates in the Southeast US. What I want to talk about now is something that looks quite different and is looking on a much broader scale. And this work really comes out of a plot that I showed previously, which was these different contributions to the alkyl nitrates that we saw observed and modeled during seekers. And if you were looking closely before, you might have noticed this sort of brownish wedge that I conveniently didn't mention. And that's because it's something that we didn't previously simulate in the model at all. So this represents very small alkyl nitrates, methyl, ethyl, and propyl nitrates that are formed from oxidation of methane, ethane, and propane. And they're not that important when you look at the boundary layer. They're a pretty small part of the picture. But when you think about the free troposphere, they, in the observations, are at least 50% or about 50% of the total observed alkyl nitrates, and maybe even more because this gray part here, this particle part, gets pretty uncertain as you move into the free troposphere. Now, the thing that makes them potentially interesting is that unlike all this stuff, which is removed very quickly, they have very long lifetimes of weeks to months. And that means that you can think about the fact that if these are formed over a continental source region where you have NOx emissions, and then they last for a few weeks before they're degraded, that could potentially be a source of NOx to the remote troposphere, and something that we've been clearly neglecting in GEOS-Chem at least. So my goal has been to try to get a better handle on actually how important is this wedge, and is this something that we need to be taking account of? And luckily, unlike the isoprene and monoterpene nitrates that are very newly measured species, we have a wealth of historical data to constrain this problem. So these species have been measured on NASA, NSF, NOAA planes for about 20 years, and we've got lots and lots of data to look at. Okay, so what I've been doing is trying to put this simulation into our model, into GEOS-Chem, in the global model at this point, because we care about the export. And this is a simplified version of the picture that I showed before, because these things are much more volatile. They're not likely to be lost much via deposition. They're not likely to be taken up into the aerosol. We've updated some of the precursors, emissions, and distributions to try to get the best simulation we can, at least over the US, where we know what they should look like, and put in the sort of standard loss processes. The one extra thing that we had to take into account for this is that methyl and ethyl nitrate, at least, are known to be emitted by the ocean. And so there's an additional ocean source, an additional straight emission of these that we need. And that turns out to be a little bit complicated to simulate at the moment. And the reason for that is that we don't really have very much data on what the concentrations of these things are in seawater. Now, previous models have just put an assumed flux from the ocean to the atmosphere. But what we're trying to do here is to use an actual air-sea parameterization to link waterside concentrations to what's in the atmosphere, so that can change with changes in temperature and wind speed and represent the seasonality that we expect. So at the moment, what I'm using is the very limited data set that I've seen published on these species, which comes from this cruise and this data by Elizabeth Dahl, where she was just in the Pacific and discovered relationships between ethyl and methyl nitrate and chlorophyll to some extent. And you can see it's quite messy. she maps these out into different regimes, and I've tried to apply those same regimes that she uses. But this doesn't let us put anything outside of the tropics yet, and as I'll show in a minute, that's a problem that we still need to resolve. So when I put her seawater concentrations based on chlorophyll and location in, we get a surface ocean concentration field that looks about like this. It's quite blocky because of these big regimes, and that's a log scale, so you can see there's quite a lot of variability there. And when this is run through the RC parameterization, we end up with fluxes out of the ocean that look like this, the plot in the middle, and that's that's on the scale of 10 to the 8, and fluxes back into the ocean in the bottom plot, which is an order of magnitude smaller. They're more or less what we'd expect because we put a lot of methyl nitrate into the surface ocean in the tropics, and we don't have any coming out anywhere else. We do a similar thing for ethyl nitrate, and it looks basically like this, just scaled down because they're lower. So this part is definitely still a work in progress. But nonetheless, I'll show you what our simulation looks like at this stage. So these are our new global distributions of methyl nitrate in the model. These are annual means, and they're distributed by different altitude regions in the atmosphere, starting from the surface up here and down to the upper troposphere. And the underlying plot is the model, and the dots at the top are a number of different aircraft campaigns that have been taken over the years. There are still more to be added here, but this covers a fairly wide range of them. We get some things right, so we have a reasonably good representation of what's going on in the tropics and that gradient from the tropics to the North Pacific. But obviously there are some big problems in terms of what's going on in the southern South Pacific and the Southern Ocean, and also potentially the Arctic, and this is something that will be coming down to that ocean parameterization. I'll show profiles in a minute that make it a little bit clearer what's going on here, but this one's methyl nitrate. If I step through to ethyl nitrate on the same scale, you can see that it's a lot lower, it's a lot less important than the methyl nitrate, and has somewhat similar distributions. And finally, propyl nitrate is the other new one that we've added. That has a shorter lifetime than the other two, and so that tends to be more concentrated over the continents than over the oceans. And we see that to some extent in the model. There appears to be something interesting going on in the Arctic with the propyl nitrate, and having spent my PhD looking at the Arctic, I'm quite curious about that, but I haven't dug into it yet at all. So to give a bit of a clearer picture of the evaluation and how the model is actually doing here, I've started comparing to a number of these campaigns using vertical profiles. I should point out that these are not point-by-point comparisons. They're just averages over the roughly right region and the right month of the campaign. Methyl is on the left, ethyl in the middle, and propyl on the right. And these plots here are designed to look a little bit at the source regions. So we expect most of the emissions, except for the ocean, to be over the continents. And for the most part, we're maybe a little bit too low relative to Seekers and DC3. But depending which campaign you look and where exactly in the US you look, we're at least reasonable for some of the ethyl and propyl nitrates. Methyl's a little bit too low everywhere, and I think that might be related to the yield that we're using in the model at the moment. I threw in frappe at the last minute because I know some people here at NCAR were involved in that. Surprisingly, despite how close it is to the sources and how hard this region is to model and how big my grid square is, we're actually doing a reasonable job with frappe as well. So if we move then to the remote areas, which is what I'm actually really interested in here, I'm using here some of the HIPPO data. So this was the pole-to-pole campaign that took place between 2009 and 2011 over several different campaigns. I've just averaged them all in together now, so I'm not looking at anything seasonal. But I've got the HIPPO data on the top, and on the bottom, the trace P data, which was about a decade earlier. And ignoring the left plots for the minute, we do a reasonable job at simulating the ethyl and propyl nitrate. And you can see that propyl nitrate ends up being reasonably high, even relatively far away from the continental source regions, and is actually in some of these campaigns is the largest contribution to the alkyl nitrates that we see. But actually, that plot looks like it might be wrong. I might check that one. But they're sort of roughly order of magnitude in HIPPO, the methyl and the propyl nitrate. The methyl nitrate is quite interesting and shows not only a difference in the model observation comparisons, but that in trace P, we were at less than 5 ppt. In HIPPO, it's closer to 10 ppt. And this could just be variability, but it could in fact reflect growth from 2001 to 2011 in the methane amount in the atmosphere that's forming methyl nitrate. And that's not something that I have in these simulations, which are all for a single year at the moment. So I think this one's probably a more accurate representation of where things stand, but we might be able to use these observations as well to map out how these nitrate burdens have changed over time. Moving to the equatorial Pacific, we see roughly the same thing as before, so reasonably good simulation. HIPPO showing a lot more methyl nitrate than the earlier campaign, PEM-Tropics B. And then moving into the South Pacific, things are just much too low. And that's the same that we would have expected from those global map distributions that I showed before. In particular, I think what's important is the shape of these vertical profiles in the observations as well. So we see these increases at the boundary layer, which suggests that this really is a flux coming from the ocean. It's unclear without more waterside concentration measurements as to what's actually in the water. But one thing I'm thinking about trying to do is to back out the concentrations that we would need in the water to reproduce the amounts that we see in the aircraft observations. So if I take a step back, what actually spawned this work is the interest in how this impacts the NOx budget and whether we have an increase in NOx caused by going from a simulation where we didn't have alkyl nitrates at all, or these small alkyl nitrates at all, to one where, although it's still a bit flawed, we have reasonable concentrations in some places over the ocean. So what I'm showing in these plots are just pure differences between those two simulations, with and without these species. They're represented as a % change in NOx, and these are over the sort of lower free troposphere, 2 to 5 kilometers, and then the sort of mid free troposphere at 5 to 8. These scales go from about -5 to 5% change in NOx. And there are a couple of different things here. I mean, by far the most obvious is that we have a new source of alkyl nitrates in the tropical oceans. That causes an increase in tropical NOx, and this is a region where the NOx is already quite low, so this could potentially have impacts on our simulation. There is some interesting spatial variability in this term that I haven't looked into in great detail. Another thing to notice is that despite the fact that I know that we're underestimating all of these species in both the Arctic and in the Southern Ocean, we do see small increases in the NOx in the polar regions, and that's also something that I'm hoping to look into more. And then the most interesting thing to me when I made this plot was these export regions, these sort of Pacific regions. It's a bit hard to tell in this light and on this color scale, but what I was expecting to see when we added these species was an increase in NOx, because we take these alkyl nitrates that we form in the continents that lock up some NOx, and we release it over the ocean in the free troposphere. What I actually see is a small decrease. And after scratching my head for a while, I think what's going on is that this represents a chemical competition between alkyl nitrates and PAN. So this is the % change in PAN over just that whole lower to mid free troposphere region. And we've basically introduced a competition both for the NOx in the source regions and for the VOCs and any flow-on species that they create. And we know that PAN is a good source of NOx to remote regions. And so basically what's happening is that we have two competing effects. We have an increase in NOx caused by these alkyl nitrates, but a decrease in the NOx from the PAN. And so they end up canceling each other out, and we end up actually with quite a small effect on the NOx in these remote areas. We can also look at the ozone impacts, because that's one of the things that NOx is really important for. And it's more or less consistent with the combination of these two plots. So The one place where we really see an increase is in that tropical ocean region, where we have an increase in NOx from these alkyl nitrates coming out of the ocean. Everywhere else, we have a pretty small decrease, and that's because the pan impact dominates a little bit over the alkyl nitrate impact. Obviously, we know that we're still really underestimating what's going on with those species in this region, so I wouldn't put any stock in this plot for the southern hemisphere, but I am hoping to get a better handle on that in the future. Okay, so this is still very much, as you can tell, a work in progress. And although I'm an atmospheric chemist, I think this comes down to having to figure out what to do about the ocean. And I'm hoping to talk to some ocean chemists about how best to try to constrain this tropical source and also the source elsewhere in the ocean. I know everyone hates it when a modeler says we need more observations, but that's basically what we need here, at least for the waterside concentrations, because without that, it's very hard to have any sort of physical understanding of what's going on with this emission flux. And what would be really great is to be able to tie these to other observable parameters, like chlorophyll, which we already have, but radiation and temperature, and have some sense of what might be going on in the ocean that can impact this air-sea exchange term. The other thing that I mentioned a couple times is that there are interesting things going on in polar regions. They may be polar-specific processes, and so it'll be important to look into the methyl and the ethyl and propyl nitrates there. So at this point, I want to leave you with most of my take-home messages, but I will warn you, this isn't quite the end of my talk, because there were a lot of sort of chemistry specifics, but I just want to back up a stage and say, you know, what is actually the point of all of this? So the first one is that these alkyl nitrates, which are important for understanding the NOx budget, they have very diverse sources. They can be biogenic or anthropogenic. They can be a whole suite of species. And what contributions you have really differs both based on your emissions, but also based on the chemical environment that you're in. And they can influence the NOx budget, and they can influence the NOx lifetime, but again, those impacts really differ from place to place, and it's hard to have a sort of globally, here is what happens when you have an alkyl nitrate. In some places where we have NOx, but not very much NOx, like in continental biogenic regions, we have VOC emissions, this formation of these species can be a significant NOx sink by locking up that NOx in the alkyl nitrate and removing it from the atmosphere. In other places where we have very little NOx and we don't have a lot of NOx emissions, like the remote oceans, injecting an alkyl nitrate from the ocean or elsewhere can be a NOx source. And really, this has highlighted to me that although we've been measuring these species for a really long time, and although their main chemical processes has been measured in labs and known for a long time, there are actually a lot of sources of uncertainty, both in their sources, but also in chemically what's happening. And I didn't talk about it at all, but the literature on the yields of these things is kind of all over the shop based on how you measure it. So there's still more work to be done, I think, in understanding these. Although the net sum on a global scale might be reasonably small, they're still important in certain local environments. Okay, so there's one other thing that I want to talk about that Louisa alluded to earlier, which is that the real reason that I'm here is working on planning for a possible experiment using the NCAR C-130. And this is an experiment called Koala, which stands for Characterizing Organics and Aerosol Loading over Australia. And I'm still hoping this can be our mascot here, although we'd have to find a big koala. So this is an experiment that we're hoping will take place in Southeast Australia in January and February 2020, which is our summer down in the Southern Hemisphere. And I just want to give you a couple of the reasons why we think this would be an exciting place to do really interesting science. So the first reason is that Southeast Australia is a global hotspot for biogenic volatile organic compounds. This is the global map of the Megan isoprene emission factors, and you can see that Southeast Australia really lights up relative to other places where we've gone in the past because of their high isoprene emissions. So relative to the Southeast US, relative to the Amazon, relative to Borneo, at least as far as Megan's concerned, we have very high potential for isoprene emission here, and that has to do with the eucalypt forests that cover this region, which we think are high isoprene emitters. It's not purely a model construct. So this is a plot of OMI formaldehyde in summer, averaged over about 10 years. And although I don't have a scale on here and I don't have the rest of the globe, you can see that the southeast part of the country here lights up, and that's coincident with where we think there are lots of isoprene emissions from these forests. One thing I'm not showing is that in addition to the isoprene, we see large concentrations of monoterpenes in this area. And in fact, one thing that's quite interesting here is that unlike most other places that we've looked, the isoprene and the monoterpenes seem to be present in roughly equal amounts. So we're used to thinking about environments like the Southeast US or the Amazon, where isoprene is really dominant. There's an interest in boreal Canada and Russia, where monoterpene is dominant. But here we seem to have some sort of balance between the two. And of course, if we look at the emission models, they don't really see that balance. They see lower monoterpenes and higher isoprene. So there are some questions around what these biogenic VOC emissions really are, and then what happens when you have such large biogenic emissions. What also makes this area interesting is that we have very low NOx, and the NOx that we do have is really isolated in these urban hotspots. So I was talking before about the Southeast US, and I was talking about very low NOx regions and very high NOx regions. But you can see that actually, there's a lot more of a smearing out of NOx because of the Northern Hemisphere background and just because there are so many point sources in that area that are able to influence one another. But when you get to Australia, 90% of our people live in the major cities. There are only a couple of those major cities, and they're spread apart. So this is the Sydney metropolitan region. This is Melbourne down here. And basically outside of that, you have pretty much no NOx except for stuff coming out of the soil and stuff from lightning. This Sydney region, where we have this very large NOx source that's isolated, is completely surrounded by national park, and that's mainly eucalypt vegetation. So we really have a very nice natural laboratory for trying to understand how this chemistry changes as you move from a low NOx region to a high NOx region and back to a low NOx region. What does the inflow look like? What does the outflow look like? Are these alkyl nitrates forming? Are other species forming that can either impact air quality in the city or go in the other direction that can impact our terrestrial ecosystems? And finally, this is a region that's really interesting from a perspective of thinking about ongoing global change. So we know that Australia has been warming quite a lot over recent decades. I imagine a lot of people heard the news story a couple of years ago when the forecasters had to add a color to their color scale because it was getting too hot in the summers. This is a little bit old, but this is the trend in maximum temperature, just overall, over this 1970 to 2011 period. And it's been basically consistent warming across the southeast. And we get some very hot days, 40 degrees Celsius in summer, usually a couple days a year. We also have extremes of hydroclimate, so droughts and flooding. And there was a very long and very extended drought across much of Southeast Australia that's finished now, but there is potential for some of these more extreme years. And this is a paper I found earlier that's showing the vegetation sensitivity to those extremes. And these places that are coincident with where we have the high isoprene emissions are also places where the vegetation is quite sensitive to whether it's a dry year or a wet year. And if the vegetation is sensitive, I would imagine the emissions are also sensitive. So again, looking in Australia might give us an opportunity to look at what the response is likely to be to really strong climate extremes, either in terms of temperature or in terms of precipitation as we move into a warming world. The last thing I'll say is that we still don't really understand what's going on with emissions and chemistry in this region. And these are some plots put together by my former student, Caitlin Lischke, who's at Berkeley now. And Wollongong is right about here. It's just south of Sydney. And there's an NDAC FTIR that's been measuring formaldehyde there for over 20 years now. And Caitlin's been looking at the trends in the formaldehyde, and we see a very consistent decreasing trend in formaldehyde at Wollongong, despite our initial assumption that with all this warming, we should be seeing an increase in biogenic emissions and an increase in formaldehyde. And in fact, if you calculate these trends on a monthly basis, that trend is pretty consistent across the entire year, with the exception of November. Now, we don't understand what's driving the background trend yet. We're still trying to come up with ideas for that. But what we think might be happening in November is that we've actually seen more warming in the local area, like right close to Wollongong, in November than we have in other months. And we think that there is an earlier onset of summer and an earlier onset of the sort of temperature trigger that you need to start isoprene emissions, and that is overlaying a background trend in decreasing formaldehyde. So there are lots of questions that we still don't really understand in this region. There are lots of interesting things that we could do with an aircraft campaign. And so with that, I'll just add one more take-home message, which is that I really do think that we can use places that are regionally interesting, like the Southeast US or like Southeast Australia, to answer questions that are globally relevant. So with that, I'd just like to thank you for your attention, and I'd be very happy to take questions.
Thanks, Jenny. For webcast questions, we're using the microphone.
So, nice talk. I had a couple, but I'll just pick one. What if you go to Koala and it happens to be a drought year or wet year? Everyone's going to say, well, what happens on the other side? So, do you really need to go twice, or do you actually hope to get a drought, or what's your sort of planning there?
Yeah, I mean, it's a great place to visit. That's an interesting question. I hadn't really thought about that. Obviously it'd be great to go twice, but I don't think that would be likely. One thing that is going to go on continuous with Koala is a ground site, and they're hoping to start measurements there very soon. And so we'll have some sense of what the variability is at the ground. If we can use the aircraft to try to connect those with the larger scale picture, then we might be able to keep the ground station running and get a sense of, if this was an extreme year, how does it vary from year to year?
I was just curious on that TDLIF disagreement you saw in SOAS. It looked like maybe there was a ground-based instrument that was disagreeing with the airborne instrument.
Yeah, that's also true.
Probably different groups or something. Can you comment on
No,
that?
they're both from the same group. Yeah, so these triangles are the SOAS sites, and then this is the average over all of Seekers. These are both from the Berkeley group, so it's really unclear still what's going on, because you can see that, the model doesn't find a difference here. Like, the model is agreeing with the ground site and not agreeing with the aircraft observations. I don't know that I have a plot of all of the different individual contributions at the ground site, but they're a lot closer. Still a bit off between the TDLIF and the SIMS and all the other things added up. But there's some fundamental disagreement here, and nobody could decide what it was. So as a modeler, I can't really comment except to say that they look different.
Well, there were different seasons, so I don't know if that would...
Oh, but they were not too far apart, yeah, seasonally.
At one point, you had mentioned that the monoterpene and isoprene contributions were of similar magnitude. So I guess there's a two-part question. How would ambient concentrations at a ground site, have those been measured? And I guess number two would be, has Alex's group in the past, because I know they've been to Australia before, measured individual tree species to maybe build that up from a bottom-up inventory?
Yeah, so those are excellent questions. So the observations that we have of monoterpenes and isoprene are quite limited, and there were a few published in a paper by Catherine Emerson from CSIRO, and that's where we get this fact that they are roughly equivalent. Those are the ambient concentrations rather than the emissions. So there have been a couple of short campaigns in Western Sydney, which is obviously not in the middle of the forest, and there was a short campaign in Wollongong, which is a little bit closer, but we only got biogenic air on, I think, 2 days. Those happen to be really hot days as well, which gives us some nice things to look at. But so they're suggesting that the ambient concentrations are quite similar. We haven't actually really measured this in in the middle of the forest, except for the measurements that Alex's group took at a place called Tumbarumba, which I didn't put on these slides. Louise has got a slide. And was it on the map?
Yeah, it's in the middle.
It's basically in the middle of the forest. And the problem with the Tumbarumba measurements, besides the fact that they were very, very short data set, is that they had very unusual conditions. So it actually snowed during their measurements there. So they're not necessarily representative. In terms of measuring things at the leaf level, they've done some, Christine would know more than I, but not a lot, and not specifically in Australia. And one of the issues that Catherine suggested with why the isoprene emissions might be so much higher in Megan than they seem to need to be for her to match the model concentrations is that a lot of the eucalypt measurements have been done on young trees, and the sort of community mean age might be quite a lot higher, which might change those relationships. So I think there's a need to do kind of all of these measurements. Like it would be great to have the aircraft to get some spatial representativeness. It'd be great to do some sort of chamber leaf level studies of what's actually coming out. And it'd be good to have the ground site to get some of the variability and how that relates to the environmental conditions. Louisa.
Yeah, thanks, Jenny. I probably just missed this, but you said something like 60% of the nitrates are lost by aerosol uptake. Was that a global number, or was that just for the biogenic regions?
That's just for the Southeast US, just the biogenics, because we didn't have the C1 through C3 in that version. And that's probably a little bit of an overestimate because we were a little bit aggressive with our isoprene nitrate uptake to match the gas phase because the particle constraints aren't quite as good. But I think that's another thing that we could get some handle on by doing sort of more of these measurements in any of these biogenically dominated regions, because there are some new measurements coming out of places like University of Washington that look at specific species in the gas phase and aerosol form.
Oh, that's all. Well, thank Jenny again. Thank you.
Thank you.